Okay, so we're now jumping a few chapters because I like to keep everything plant related together. So we're going to be jumping now to chapter 34, and then we will be coming back afterwards to the plant chapters. So fungi, which is what we're going to spend our time talking about now, are a very abundant and diverse group of eukaryotes. And let's classify them in terms of terms we've discussed so far. So they're heterotrophs. This means that they absorb organic molecules, right? Carbon. And also they do this for energy as well through their cell walls. And simple molecules are going to pass easily across the cell wall. However, one way that they do a great job of decomposing a lot of material in our environment that needs help being broken down is that they're able to secrete enzymes and break down molecules in their environment. And for this reason, they play a very strong role in recycling a lot of dead material in our environment. So they decompose a lot of no longer living land and plant and animal tissues. And a lot of this is going on when you don't even notice it. A lot of fungi live in the soil. And again, they have a really strong role in cycling carbon because they're able to do a lot of decomposition. They're able to break down molecules that a lot of other types of organisms are not able to break down. And we'll talk more about those types of molecules. But for now, let's just say some complex molecules in bulky tissue in our bodies and plant bodies that need to be broken down, particularly in trees. Fungi have the enzymes necessary that they can secrete and then decompose this organic matter and then restore that carbon back into the environment in a usable form. So this question, actually, I haven't introduced the term hyphae yet. We'll talk about that on the next couple slides. It's just a structure that is what helped make up the fungi, but really here looking at surface area, volume ratios, and we'll talk more about this in class. So now we're going to talk more about these hyphae. You can see them in this image here. So fungi are able to really gain access to a lot of nutrients in the environment by continually growing and reaching for new resources in other areas. So most fungi are going to be multicellular, and again, most of them are going to have this branch structure that we're looking at here, known as hyphae, and it gives them a really large surface area for absorbing nutrients because it does form these long, narrow structures that have great surface area and allows them to take up nutrients through that surface. And this growth in terms of finding nutrients is important because they don't move. So they gain new access to areas of food by growing, but they're not able to physically move. And in some ways, this is similar to what we see for plants, and we'll talk more about them later on. So when resources are really high, they're going to just keep growing rapidly. When resources are low, then they're going to want to maintain their energy, slow down that growth. But during high times of resources, they can really branch out, and that will allow them to then be ready to go whenever resources get low. So again, I said that fungi also have cell walls similar to plants. That was that typo on the previous slide. And this does help them and prevent them from busting open because if they're exposed to a very dilute solution, again, remember what we talked about in terms of concentration. So if you have something that's, let's say, concentrated on the inside and not so on the outside, then what happens is that in order to balance concentration gradients, and we'll talk more about this later on, if the solutes aren't able to move across the cell wall of the fungi, then water is able to move, and then water will enter the fungi in order to dilute the internal solute concentration so that it's equal to what's outside, and that would, if there was no cell wall, cause it to burst, but because there is a cell wall, it prevents rupturing. There also is continuity, and this is important, between the individual cells. So if fungi are made up of these long, stretchy hyphae that are able to reach out into all sorts of areas, imagine you reached out into an area and you have food source, but then way back behind you somewhere there isn't any food. The fact that all of these cells are connected and have continuity allows for them to pass nutrients from nutrient-rich areas to nutrient-poor areas. And so there is continuity between these cells. You can see here that even when later groups started having septa, which is a cell wall that separates individual cells, they still have a pore that allows for movement between cells. So that's really important, the continuity. It allows them to maintain growth of these very large structures. And we're going to see that they are also above ground as well as below ground. And again, these early diverging groups prior to the septa, it was just one giant cell with many nuclei, but later on, even when the septa eventually became part of fungi later on in evolution, and this is important because it gives you strength and allows for, for us, let's just think more about strength, but again, the important point here is that they still have a pore, and that pore is going to allow for movement of nutrients. So an exception to the rule for this hyphae type structures that you see are yeast. Yeast are a form of a fungi, and they're single-celled, as you're seeing in this picture, and they reproduce by budding. So you can see these little buds that are breaking off. Here's another bud, and that will form a new cell from the original. There's evidence that they are descendants of yeast that actually produced hyphae, but that in the yeast they were lost over time. And the reason for that is that yeast tend to be prevalent in very nutrient-rich environments, and so they don't have the same need for hyphae, and so that particular structure was lost over time. Although in some instances you do see some hyphae-like structures, but it's not what we tend to think about with yeast. They're very common on the surface of plants, and you might think of them in your day-to-day life as being important for alcoholic beverages and the production of bread that you might eat. Again, I mentioned that one of the major roles of fungi is that they're able to decompose land, plant, and animal matter that other organisms are not able to break down. So really, we would be looking outside, seeing piles and piles and heaps of dead trees everywhere if it weren't for fungi that are able to break down that matter and then return CO2 into the environment. So it's very important. They have a very strong role in carbon cycling and returning nutrients to the soil. Again, they move very well through the soil, so they can get access to lots of different nutrients that need to be broken down. And again, the thing that they're able to break down, and we get to plants later on, we'll talk about this, but in the bark of a tree, plants have, in particular in the bark, you see lignin, which helps to give you structure to the bark of a tree. But lignin is incredibly difficult to break down. Some bacteria are able to, but really most of the time, if you think about the bark of a tree being broken down or the wood of a tree being broken down, that woody structure is mostly being broken down by fungi. We'll look at this in class together. So remember that fungi, hyphae, the hyphae of the fungus, it is adapted to basically grow and seek out nutrients. So you already have a structure that is seeking out nutrients, and then you add that into, let's say, a plant that has a cut in it, and this really allows for a fungus to be well adapted to enter via wounds and cause a lot of destruction to a plant in particular if it infects. So we think of plants as being extremely vulnerable to a wide variety of fungal pathogens. In terms of humans or vertebrates, even in broader terms, we don't think of them as being a major infectious agent. However, you might have heard of this one exception that's been very prevalent in the news recently. North American bat populations have been threatened by a fungus, a white nose. That you might have heard this, bats with a white nose, this disease. And a lot of this, actually there was a person who came on campus to talk about this a while ago. If you're curious, you can read more about this, but this particular type of fungus tends to infect hibernating bats. If they were awake, then it wouldn't be a problem. They would clean themselves off, the fungus would be gone, it wouldn't be a problem. But this fungus tends to infect when the bats are in hibernation, and by the time they actually wake up, too much damage has been done, and it's been a major threat to the bat population in North America. And people are still studying that particular phenomenon and ways to stop that from continuing. In humans, most of them aren't really considered life-threatening. You might get athlete's foot, yeast infection, but nothing too serious. Also, you have insects and other invertebrates that can be susceptible. Some get trapped because hyphae can form these sticky traps. So you can see here a worm being trapped in the soil. But really, we don't think about them very much in terms of our own lives other than being a little bit annoying. However, it should be mentioned that sometimes they can be difficult to treat because since they are a eukaryote, it's tough to find differences between them and us in terms of treating those infections. So it's not like you can just give an antibiotic, they don't work on a fungus. Well, so we know that a fungus can be detrimental to a plant if there's a cut, but it also can be very helpful if there are mutualistic relationships between the fungus and the plant. And in fact, in many instances, these fungi are crucial to the life of the plant because it helps to provide nutrients to the plant, since the fungus can reach out into the soil in much further away than where the roots might actually travel. And in exchange, the fungi is happy because it gets lots of carbohydrates from the plant. So certainly this is a very mutualistic relationship, and you see this in a variety of different instances as being crucial. In fact, there have been studies where you remove the fungus and the plant's not very healthy at all. You don't tend to see these symbiotic mutualistic relationships between fungi and animals. Sometimes you see them with the fungi and insects, but we're going to look at this particularly here for plants as being very critical to their function of both of the organisms. In addition, we don't think about this as a land plant, but we know algae is an early type of photosynthetic eukaryote, a predecessor in terms of an evolutionary history from land plants. And green algae oftentimes can be found in regions where not a lot of organisms can grow, but if you have a green algal cell working together with certain types of fungus, then you can form a stable complex, a stable association between the fungus and the algae, and that complex between them is referred to as a lichen. And these can be and not only green algae, but also cyanobacteria, pretty much anything that's able to create carbon, and then that reduced carbon can be used by the fungus, and then the fungus is also able to provide nutrients that it takes up from the environment. So the two tend to work very well together, and they do well in regions where other organisms can't exist. So for example, you wouldn't necessarily think of, hey, on the side of a tree would be a great place to grow, but these types of structures can grow very well in these tough to live in areas due to the ability of them to work together to gain nutrients and carbon from the environment. So now we're going to talk about a mechanism that we haven't discussed yet, but it's going to become very important to remember later on when we talk about plants. So I don't want you to forget about spores. And this is true for what we're talking about now, and spores is true for fungus, but it'll come back and be very similar when we look at plants later on. So fungi, we know that they branch out, the hyphae grow, but sometimes, you might reach an area and they're just out of luck. You can't find any more nutrients, but they don't want to cease to exist. And when this happens, they can send out structures known as spores. And these spores can be carried by wind, water, animal to faraway regions. And once they reach those regions, they can then encounter new nutrients and regenerate that life once again. They tend to be encased in a very thick protective wall, which is important because they have to be able to survive and in order to then lead to the next generation. So fungi will produce huge numbers of these spores to enhance survival. If you produce enough of them, someone's bound to land somewhere and do well. They can be viable, depending on the species, from a few hours to many, many years. And again, it allows these fungi to use patchy resources. So oftentimes, again, if resources become very limiting, then it triggers spore formation, which allows that organism to then reach out and recultivate in new regions. They can be formed in two different ways. They can be done sexually or asexually. Sexually, we know they're produced by meiosis. Mitosis, it would be asexually. And I should mention it'll probably be on a future slide, but spores are always going to be thought of as haploid. So this is the fungus you're probably most familiar with, right? Your everyday common mushroom. So mushroom is what we'd consider a fruiting body. It is actually a fungus that is produced as they have hyphae that's part of their structure. So if you're eating a mushroom, you're eating lots of little hyphae. This is what you're seeing here. And not all fungi will produce these fruiting bodies, such as what you're used to looking at in the forest seen here, but a lot of them do. And they will produce spores, and we'll talk more about that. And many will actually forcibly eject those spores to allow for regeneration in other regions. And we'll go on from there. We'll talk about these much more. All right, so those life cycles we talked about before, the haploid dominant, diploid dominant, we're going to get back to that right now. So all those terms I taught you before are important. This particular life cycle, fungi are usually haploid, so this is going to be very similar to the haploid dominant eukaryotes that we looked at previously, and they're going to alternate just like all eukaryotes do that undergo some form of sexual reproduction. It's going to alternate between the sexual cycle and the asexual cycle. First, we're going to look at the asexual cycle. So remember, since they're haploid dominant, this asexual cycle will produce more haploid organisms because the dominant form here is haploid, and that will allow for the dispersal of spores, which are haploid. And in this case, again, your mycelium are made up of, this is haploid because it's haploid dominant. And then on the next slide, we're going to look at how the sexual cycle works, and then we'll put the whole thing together. All right, so this is a big old cycle, so we got to find some place to start here. So let's look at where we left off in terms of spores. So if you have a spore that has landed somewhere and it's haploid, and it's going to germinate. So now we're going to grow new mycelium, right? So now we're going to get all those hyphae branching around. And then what can happen at some point is that you're going to have two hyphae that connect to each other. So here's hyphae number 1, and here's hyphae number 2. And they're going to fuse. And when they fuse, the cytoplasm is going to fuse, and you're going to reach a state known as the heterokaryotic cell. And in this cell, let's see if I can draw this for you, it's a little bit of each type. So let's say that this one is then going to, let's say this is a cell here, right? So we've got lots of cells with septa separating them, and now we've got this other one, and now they're going to fuse together. And I wasn't drawing the nucleus for all of these, but this one has a nucleus, right? And this one has a nucleus, and these two just fuse together. So this new cell here, which is the result of fusion of the two together, that's called heterokaryotic. So the nuclei are still separate. So here, I'll draw some nuclei in the other ones so you can see the nucleus in the other cells. But what's interesting in this heterokaryotic cell is that the cytoplasms have fused, but the nuclei have not. Right, so heterokaryotic unfused nuclei from different parents. At some point after that state, then you fuse the nuclei. You're going to then have the diploid state, and then immediately they're going to undergo meiosis and reproduce the spores. This is actually something that's very unique about fungi, and I believe this is all on my next slide, but this heterokaryotic state is something you don't generally see. And the reason that you don't see this is usually you have fusion of cytoplasm and nucleus all at the same time. In fungi, these are separated. So note that it's really important to note this, that step 1, you fuse the cytoplasm and then you hang out like that for a while in this heterokaryotic state. After that, then when the appropriate signal comes, then you fuse the nucleus. So again, these are separated distinctly in terms of time, and that's an important point that is unique for the fungus. So everything I said in the previous slide is listed here. So again, they undergo sexual reproduction, and we know it's sexual because you're going to have fusion, right? This portion is fusion, you become diploid, and then you restore the haploid number. And again, this is a unique feature where the fusion is not immediately followed by the nuclei fusing, and that leads to this heterokaryotic state because there's a pause after the cytoplasm fuses before the nuclei fuse. And you should know these terms: plasmogamy, karyogamy, and then heterokaryotic is when you have the two unique nuclei in one given cell. So depending on where a given fungus falls on the evolutionary tree, they might spend more or less time pausing between plasmogamy and karyogamy. The earliest fungi will pretty quickly fuse the nucleus. After plasmogamy, you don't see this heterokaryotic state. Some do have this heterokaryotic stage with many haploid nuclei, so you could maybe have a lot of them that are going to be like this, and then others have plasmogamy followed by mitosis. So that would basically be taking that heterokaryotic cell with the two unique nuclei and then rapidly reproducing them so that you have many, many, many of those heterokaryotic cell. In those instances, we call these dikaryotes, and about 98% of all fungi fall into this category, and we'll talk more about them later on. We'll look at this in class together. And I just want to point out that when I showed those hyphae tips fusing to, again, have the fusion of the cytoplasm to get the heterokaryotic cell, that doesn't happen just randomly. It is based on gene type. So unlike in some organisms where you have males and females reproducing together and having sexual reproduction, in fungi we don't have that. Instead, what you have are no male and females, but you have mating types. And even though two fungi might look very much similar, they might have different mating type genes. And so what you're looking for to increase diversity is having fertilization where you have different mating type alleles. And that is something that allows for fusion. If you don't have different alleles, then you can't have fusion. The more alleles you have, obviously, the more likely you are to encounter a mate. So having more alleles is better because that allows for more opportunities for fertilization. And as an example, the common mushroom can have up to 20,000 alleles, and that allows for them to very rapidly reproduce. Just realizing there's a term here some of you may not know. So just making sure allele is just a fancy way of saying version of a gene. So there's a gene that codes for mating type, and you need to have different versions of that mating type gene in order to allow for reproduction between two individual cells or two individual hyphae. So I always get lots of questions on this particular mechanism, and to be honest, I've done some digging and I can't really give you an exact answer as to how this works, and it still actually manages to get the right chromosome number back. But I will briefly describe something that's unique to the fungus. About 20% of fungi do lack sexual reproduction, and to gain diversity, they have a process known as parasexuality. So parasexual species generate diversity by crossing over of DNA during mitosis, so exchanging bits of DNA between chromosomes during mitosis. However, they do not undergo meiosis, and there's a series of events that take place, which you can see here. So if we walk our way through them, step 1, you have the hyphae fuse and you get the heterokaryotic cell. Step 2, the nuclei are going to fuse. You see this here, and they're numbered over here as well. And then step 3, instead of going through meiosis, these cells actually just gradually lose chromosomes until they restore the original chromosome number. But you do note here that crossing over has occurred. And this is something I talked about last semester, so if you don't fully understand this, don't worry. But crossing over, if you can visualize, it looks pretty straightforward. You have two different cells with similar with the same genes but different versions. So here you have the one in dark blue, here it's in light blue, now you've got both in the heterokaryotic. And then what you see is crossing over allows for mixing and matching. So now you can see here there's some combinations of the dark blue and light blue. You see that over here as well. That allows for new combinations to be formed, gaining diversity. You still see them here, right? This one is a new combination. This one's a new combination. And then they restore the original chromosome number through mechanisms that I don't believe are fully understood at this point. But you'll see that there is no meiosis happening, yet they still are able to gain diversity and restore the haploid state. So if we just briefly look at the phylogenetic tree of fungi, you can see here that they're cousins to the animals. So here we have animals over here, and then you're going to have the fungi, and you can see that one of the earliest traits that had to evolve in all fungi is chitin. So chitin in the cell wall helps to give it this strength that allows it to not, or prevents it from bursting if too much water flows in. So that's true for all fungi. Later on, you see hyphae come on the scene as being very important. Then you see septa, right, that allows for individual cells to have their own separate processes, but still having connectivity between the cells. And then you also see that these complex multicellular fruiting bodies evolved twice, and these are found within a group of fungi known as the Dicaria, which is where most of your fungi are actually found. Most of them are going to be in this group. And Dicaria are going to be ones that have many cells in that heterokaryotic state, and we're going to look at some examples of that later on. We'll look at this in class together. So similar to when we talked about eukaryotes, I don't expect anybody to memorize all these little facts. I'm just giving you some examples of different types of diverse fungi. So you can see that the earliest fungi in the evolutionary tree are ones that are unicellular. They do not have true hyphae. They're mostly going to be aquatic or in moist environments. They lack that heterokaryotic stage. So again, in these early fungi, you very rapidly went through plasmogamy and karyogamy, so you never even had that heterokaryotic state. And a few of them are pathogenic, but mostly they're just going to decompose organic compounds in their environment. So now moving on to another group, you can see again that this is where hyphae are first found. So they do have true hyphae. They produce spores. Now you're going to see that we're going to just separate plasmogamy and karyogamy. So there will be that heterokaryotic state, and they're able to produce spores. And septa are found here, but only in the reproductive structure. So you see some septa, but it's not throughout the entire organism. So this could be sort of the starting point for allowing for the spread of that characteristic later on, right over here, through all fungi at a later date. Because we know that some characteristics might be used for one reason in an earlier group, but then over time it might be adopted for other purposes, such as now we see regular septa throughout the organism. So this next group are just worth mentioning because we talked about the importance of the symbiotic relationship between a fungus and a plant, and these are the types of organisms that do that. So they do associate with plant roots, and they become very important for allowing both the survival of the plant and the fungus. So they have a very strong ecological importance. So now when we talk about the Dicaria, this is again where most of your fungi are found, 98%. And what's important about the dikarya, remember di means two, so I underlined it here for you. So when you have fusion of two cells after plasmogamy, right, you can have a cell, let's see if I can draw this, you can have a cell with two nuclei, right? So this is one giant cell with two nuclei. It's the two is important, and they're able to limit it to the two nuclei because septa will be formed after each round of mitosis so that it stays as two. So dikarya, what's unique is that after this heterokaryotic stage is reached, they just keep undergoing mitosis. So you can see that maybe right afterwards, they're going to produce more of the same thing, and this just keeps happening over and over. So you end up with a giant structure made up completely of these dikaryotic cells because they keep undergoing mitosis with septa between them, so they stay in this 2-nuclei state. So the dikarya is. In this group, you're going to find all sorts of different types of organisms like mushrooms and yeast. And again, yeast we think lost some of these characteristics later on because we don't think of them as having hyphae, but they are considered part of this group. And they all share very similar life cycles, but they evolved the fruiting bodies independently. So we're going to talk about the two subgroups, and you can see them here to your right, that it evolved twice independently. So there must have been some beneficial reason for this to see it happen twice. So you see it here and you see it here, and they both work in similar forms, although we'll see some differences in how they reproduce. So we're going to look at two different types of these dikaryotic organisms. We'll start with the Ascomycetes and then we'll move on to the Basidiomycetes. No one needs to memorize this. I just think it's useful to look at this particular life cycle to get a feel for how it's distinct from the general one we looked at earlier. And in some fungi, this can get very complex. We're not going to get into that. So let's start. See, they always start with number one. All right, we'll start with number one over here. So you have hyphae in the environment, right? And they're absorbing nutrients, and then they're going to fuse. And you can see plasmogamy took place here between two distinct mating types, right? You need two different alleles. And if you were to zoom in over here, you could see the fusion between those two cells. And then once that happens, you're going to have reproduction of those dikaryotic cells so that you get more and more of them. And that can be seen here, right? Lots of these dikaryotic cells that are being produced. Eventually, some of them will differentiate to release spores and restore the haploid state. So you can see here that these structures called an asci are formed, which is why this is called the Ascomycetes group, after the structure that forms. And once this forms, then you can see that they undergo karyogamy. So if you were to zoom in on this cell here that's dikaryotic and then zoom in after they undergo karyogamy, you can see they have a diploid nucleus. Then they undergo meiosis, and then they're eventually going to lead to 4 haploid nuclei, and then eventually they undergo mitosis to give you some more nuclei, and then those spores are going to be released and the whole process starts over again. And if you look at this particular structure, again, the hyphae are going to be what comprises that fruiting body, although if you look at this fruiting body, the haploid cells actually produce most of the fruiting body, and the dikaryotic hyphae only produce some of it, right? So the haploid hyphae are most of what you're looking at here, and the dikaryotic hyphae are just a few of what you see in the fruiting body. So this is a similar life cycle. Again, just a few differences here based on structures since they did evolve independently. So Basidiomycetes, you see a different structure that eventually undergoes karyogamy, but let's start again with number one so we can just review the basic life cycle, which is important, right? So we're reviewing some points here, two different mating types. You get plasmogamy, so you have fusion between two cells, leading to the dikaryotic state that undergoes mitosis. So you can see that there are now many of these that have been reproduced. Unlike the Acidomycetes, the Basidiomycetes is, or actually unlike the Ascomycetes, the Basidiomycetes, the fruiting body is pretty much 100% dikaryotic. So all of that fruiting body is going to be made up of these dikaryotic hyphae. And then if you look, this is sort of like those traditional mushrooms you're used to looking at with the gills underneath the mushrooms. Those gills are lined with these basidia, which is where the name Basidiomycetes comes from. And then if you look in there, you then have these specialized cells that will eventually undergo karyogamy. They're going to fuse to give you that diploid state, just like we looked at for the generalized life cycle. Once they fuse, you then undergo meiosis to restore the haploid number, and then you're going to have dispersal of your spores, and then you start all over again. So the life cycles are both of this, of the Basidiomycetes as well as the Ascomycetes. Both of them have a lot of similarities for the basic life cycle we looked at previously, but obviously the dikaryotic state is reproduced via mitosis in both of these types of the Ascomycetes and the Basidiomycetes. So that's an important distinction. But other than that, just look at the similarities between. I'm not going to ask you to draw this whole cycle out, so don't worry about that. So again, I really want you to get the basics for fungi. So they're heterotrophic. We know that they're able to decompose land plants and animals. They're important for recycling carbon. The role of hyphae and spores in their life cycle. That life cycle, the generalized one, you should know, and the unique features that go along with it: plasmogamy, karyogamy, heterokaryotic state. And then a basic understanding of the phylogeny and diversity. I'm not going to ask you a lot of details, but you should have a basic understanding that the cell wall had to come first. So you needed to have chitin as the first step, and then later on septa are going to form. I'm not going to test you on any of their details, right? You got enough to learn between the life cycle and the basics of what a fungus looks like and how it interacts with its environment.